We discussed earlier how different gene variants can give increased risk of particular diseases, and the risk can be further exacerbated by environmental effects like smoking. To provide some clarity on how smoking can affect genes, we will visit the subject of epigenetics. You may remember that I mentioned epigenetics in my first tutorial. Here is a quick recap on what we discussed. Epigenetics is the study of changes in gene activity caused by factors that are external of the DNA sequence. Epigenetic regulation is what controls the spacing between heterochromatic and euchromatic regions of the genome, and hence which genes are expressed in each particular cell type. With all this in mind, in a muscle cell, genes that code for proteins to allow muscle contraction will be highly expressed, while in neuronal cells, muscle protein expressing genes will lie within the heterochromatic regions of the DNA, as there is no need for these proteins in such cells, and hence are suppressed. The most commonly studied example of epigenetic regulation is DNA methylation, whereby an enzyme classified as a writer adds a single methyl group to each cytosine in the DNA sequence. This recruits a protein known as a reader, which can cause further epigenetic events that modify histones. Overall, this leads to the pulling of the histones to cause a tight heterochromatic conformation. The rate of DNA methylation across the human genome shows trends throughout the course of a human's life. This is most evident during embryonic development. You can see that at the time of fertilization, the paternal and maternal rates of methylation are different, and at this time, both rates decrease to minimal levels. And at the time of the formation of a morula, the two rates converge and increase at the point where the blastocyst has been formed. And throughout the course of the fully developed human's life, the methylation rates decrease progressively. Epigenetic regulation is external to the genome, and so it is said that an organism's epigenome is the buffer between its stagnant genome and its variable environment. This relates back to the question of whether a complex disease can be attributed to an organism's environment or a particular genetic defect. Epigenetics provides some clarity because the environmental conditions such as diet, lifestyle, and smoking can affect the epigenotype and hence the gene expression profile, thus affecting the organism's propensity towards a disease. It is known that the risk of disease increases with age, and genetic diseases can be correlated with aberrant methylation pathways. Notice here in cancerous cells that there is a rapid decline in the rate of DNA methylation between the paternal and the maternal genome. CpG islands are lengths of DNA constituted by repeats of cytosine followed by guanine. Statistically, CpG islands are very likely to show a high rate of methylation at the cytosine residues. CpG islands are often associated with the regulation of genes. This is because they lie within or adjacent to the promoter regions of the genes. The exact mechanism of regulation will be discussed later in the RNA world hypothesis chapter. Genes that are regulated consistently through organismal generations are called imprinted genes. The purpose of this being to pass instructions for gene regulation from parents to offspring. Overall, this graph depicts how parental epigenetic identity is regained during embryogenesis, and how the methylation levels stay relatively constant throughout the adult human's life. Furthermore, in the event of cancer, spurious changes take place at a global, at the imprinted genes level, and at the CGI islands level of methylation. Another factor that needs to be considered in an individual's propensity towards disease is their enterotype. This term describes the microbial composition of organisms inhabiting the individual's digestive tract, primarily the small intestine. This field of research is known as microbiomics, and it studies the effect that the population of microorganisms living in the gut have on the host's health, both long-term and short-term. We see here how the enterotype of an individual affects the epigenotype. A high level of what are known as bifidobacteria increases DNA methylation in the host because the food that passes through the gut is metabolized by bifidobacteria, which produces large amounts of folate, a vitamin that promotes DNA methylation. Meanwhile, a high population of firmicutes promotes a different type of epigenetic regulation, known as histone acetylation. A high bifidobacteria composition is generally considered healthier than firmicutes. The plasticity of something refers to how easily it is changed. The epigenotype and enterotype of an individual are most plastic during the early stages of life, and they become more rigid as the individual progresses towards adulthood. Hence, it is important that a mother maintains a good diet while her offspring resides in her womb during pregnancy, and that the child is given a good diet from an early age to give minimal risk of disease in later life, as these two factors will go on to affect the individual's metabolism. Different enterotypes and alterations in epigenetic activity at specific regions of the genome can be mapped to specific diseases. The location of these changes in epigenetic activity with regard to specific diseases requires further study to determine the exact cause or defect.